gradient. Does that make sense? Okay. Not everything gets to move through the cell. Some things have to go by way of protein. They have to actually be allowed to flow into the cell. Why would this be important? Anybody else? Let's see if anybody else has some ideas on why it would be important for materials to be able to move into the cell.
Because the cell may require something, but you don't want to just
You don't want to have
the wrong thing going in.
We don't want the wrong thing. It's selective. The cell might need something. Sometimes it's easy for a material to flow and get into the cell and the cell not have to use energy. So some things get to move simply because of their concentration gradient. Sometimes the cell needs control and it will have to expend energy to get that material. So that is why when we look at gradient and flow, that they are important. And that plasma membrane and the nucleus of the cell and other parts, they can be working to either keep something in or bring something out, or keep something out or bring something in. And then sometimes, if we just have too much of a pressure gradient and it can just flow freely, if it's too much pressure out here, it can move in here. And if it's not enough pressure, we can go back and forth for pressure gradient. Okay? So that's sort of part of what's left over in the lab. So, but we'll definitely get more into that in the other chapters. So let's take our break, be back at 9:30, and we're going to work on lab 1.